Bismillahirrahmanirrahim. Assalamu alaikum. Dear students, in this video we will learn about DNA transcription. The contents of the video are introduction of transcription, requirements of transcription, functions of RNA polymerase, process of transcription, post-transcriptional modification. We know that DNA is known as hereditary material, and this DNA is passing from generation to generation. Means this DNA contains all the information of the organism, all the characters, their expression is present in the DNA molecule, and how the DNA molecule, which is the genes, how they are expressed. So this process is taking place. The DNA, from the DNA molecule, RNA are synthesized, and then from the RNA proteins are synthesized. So this whole process is separated into two steps. There are two steps of this process, from DNA to RNA and from RNA to protein. The first step is transcription. So in the transcription, DNA molecule is working as a template molecule from where the RNA is synthesized. So transcription is the process where from the DNA RNA molecules are synthesized. Then the next step is translation. Translation means the synthesis of protein sequences of amino acids from the RNA molecule. Transcription is taking place inside the nucleus, while translation is taking place in the cytoplasm outside of the nucleus in eukaryotic organisms. While in prokaryotic organisms they do not have the nuclear membrane, so both the steps are taking place at the same area. What is transcription? First we have to understand that transcription is the synthesis of mRNA from a DNA template. This means that the DNA is actually working as a template. The parental DNA is working as a template from where the RNA molecule is synthesized, and this process is taking place from 5' to 3' directions. So this is the transcription. Transcription is the process of synthesis of RNA molecule from the DNA molecule. During the process of transcription, a DNA sequence, means the parental or original DNA sequence, is used, is read by the RNA polymerase. RNA polymerase is an enzyme, a special type of enzyme that is synthesizing, producing RNA from the DNA. So we can say that transcription is the first step leading to gene expression. What are the requirements of transcription? The process of transcription has some basic requirements. There are many requirements, but at least basic requirements, the primary requirements are template, means the DNA molecule from where the RNA will be synthesized. So this primary or this parental DNA molecule, which is working as template. Then we need substrate. Substrate means free nucleotides These free nucleotides will be available in the environment of the cell, from where they will be picked up on their requirement. For suppose adenosine triphosphate, guanosine triphosphate, cytosine triphosphate, and uracil triphosphate. These are free nucleotides. They are present. They are working as substrate for the synthesis of new molecule, means RNA molecule. Then we have the requirement of the enzyme, and this enzyme is very special for this process. This is known as DNA dependent RNA polymerase, or shortly it is also called as RNA polymerase. So this RNA polymerase is a very special type of enzyme which is working for the transcription. Then we should know about the functions of the RNA polymerase. What are the functions of RNA polymerase? The RNA polymerase actually recognizes the promoter region. What is promoter region? The promoter region is the important region of any transcript, or the portion of the DNA which is going to be transcribed. This portion contains some basic segments of nucleotides, and from this site the enzyme starts to read the DNA molecule. So this is known as the promoter region. Then the other function of RNA polymerase is melting of DNA enzyme. Means there are two other enzymes, topoisomerase and helicase enzyme. So topoisomerase is uncoiling, the supercoiling of DNA molecule, and helicase enzyme which is breaking down the hydrogen bonding of the DNA molecule. In the result, the two DNA strands get separated from each other. Then other function of RNA polymerase is RNA priming. So this is actually finding the area, the place from where the enzyme has to start the work. Then the other function of the RNA polymerase is RNA polymerization. Means the new and free nucleotides which are present in the environment of cell, they will start to aid on the DNA template. And in this way, these are the important functions of RNA polymerase. Also the recognition of termination sequence, means that when the enzyme is reading the DNA molecule and it has to transcribe a small segment of the DNA, after that this process has to end at a point that is known as a termination point, terminator sequence, where the transcription finishes. Then we should know about the process of transcription, that the process of transcription is divided into three phases, three stages. So three steps. These three phases or stages are initiation phase, elongation phase, and termination phase. What is initiation phase? Initiation phase means RNA polymerase recognizes the promoter region and starts the transcription. Means in initiation phase, the process of transcription is started. Then the second step is elongation phase. In the elongation phase, the RNA polymerase continuously adding the new free nucleotides and the chain of RNA is continuously growing. This is known as elongation phase. Then there is a termination phase, means the RNA polymerase slows down and stops the adding of free nucleotides. So the process of adding of nucleotides for synthesis of RNA molecule is stopped, and the nascent RNA molecule, which is recently synthesized over the DNA molecule, so this RNA molecule will be separated from the DNA template molecule. So we have to learn each step of the transcription both in prokaryotes and eukaryotes. So first in prokaryotes. So we have to learn what is promoter region. The promoter region is the region which is present at the beginning of the transcription site, or we can say if one or two or few genes have to be transcribed, so each gene or group of genes that must have some promoter region in their beginning from where the RNA polymerase should start. So promoter region and is recognized and the sigma factor binds to it. So what is sigma factor? As we have learned that the polymerase enzyme, RNA polymerase enzyme, consists of different subunits. So like alpha subunit, sigma, and so on. So what is happening? The promoter region is recognized and sigma factor of the RNA polymerase binds to it. Then some proteins are also used in this process. These are called as transcription factors. These proteins also binds to the promoter region of gene for the starting of process. So in case when there is appropriate transcription factor and RNA polymerase binds to the RNA polymerase binds to form an initiation complex, means the structure that is known as initiation complex, where the transcription factors, transcription proteins bind and RNA polymerase bind there. Then RNA polymerase melts the DNA. As we have already discussed, the two enzymes are used, topoisomerase and helicase. So the two enzymes melt the DNA molecule, then they create the transcription start site. So when this all is happening, the polymerization of RNA begins. So prokaryotic promoter regions are specific or conserved sequences which contain some sequences like Tata box or Pribno box. These are actually conserved sequences of nucleotides which are present in the promoter region of prokaryotic cells, prokaryotic DNA. So for suppose 10 base pair Tata box or 35 base pair Pribno box, these are the specific conserved sequences in the promoter region of prokaryotic cells. RNA polymerase binds at this area of promoter region to make closed complexes. So this structure, this diagram shows us the transcription start site. This is the site from where the process of transcription starts. So this region will be transcribed. So before this region we have some conserved sequences like 10 base pair region known as Tata box and 35 base pair region Prevno box. This lower one is the template DNA molecule and this upper one is the coding strand, or we can say this is the newly synthesizing RNA molecule. So from this point the process of transcription will start, and here is the termination signal, termination site. So the enzyme will run from here up to this region. When the enzyme reach it here, and it will get the termination signal, then the process of transcription will be stopped and the newly synthesized RNA molecule, which is shown here, this will be separated from the template DNA molecule. Now we learn about eukaryotic promoters. Each type of RNA polymerase uses different types of promoters. For suppose promoters used by RNA polymerase 1 and RNA polymerase 1, 2 are same as prokaryotic DNA molecule, while promoter used by RNA polymerase 3, this is totally different from the promoter of prokaryotic DNA molecule. So this promoter region contains a Goldberg-Harkness box. This is 25 to 30 base pair. This is also known as Tata sequence or Tata box. Then it also contains a CAAT box, which is 70 to 80 per base pairs. So we can say the eukaryotic initiation process is very complex in compared to the prokaryotic initiation process. Here are the sequences. These are shown. We can see this is the start site, and before this here are the segments, conserved sequences. They are known as promoter region. So the promoter region contains Tata box, which is also known as Harkness box, CAAT box, and GC rich box. So these all are called as cis acting elements, and 6 acting elements here are as promoter region of the eukaryotic DNA molecule, and here are the structural genes which will be transcribed. So the process of transcription in eukaryotes will start from here, from this point where the promoter region ends and the structural genes start. Then we are learning about the second phase, which is known as elongation phase. So RNA polymerase binds at the promoter region and making a pre-initiation complex. Then a conformational change takes place in RNA polymerase. So the RNA polymerase starts its process. The first nucleotide almost always in many of the cases is a purine associated on a beta subunit of the enzyme. So the beta subunit of enzyme will associate with the first nucleotide is a purine. RNA polymerase catalyzes the formation of phosphodiester bond in presence of appropriate nucleotides. So we know that there are free nucleotides present in the cell environment. RNA polymerase will form the phosphodiester bonds between the chain of new connecting nucleotides. In this way, the RNA chain is elongating in 5 to 3 direction. Now, third phase of transcription, which is termination phase. So termination phase, in termination phase, RHO dependent. What is RHO? It is a type of proteins, and these proteins binds to the RNA transcript or RNA newly synthesized chain of RNA, which is now in the free, which is already synthesized. So what is happening? RHO protein will bind over the RNA transcript, means the RNA molecule which is synthesized over the DNA molecule. So RHO will bind over and run throughout and finally will come to the termination point. So this will slow down the process of polymerization of new nucleotides. So the RHO will separate the RNA transcript from the DNA molecule. Bacterial RNA polymerase sometimes recognizes DNA encoded termination signals and dislodges. Means in bacteria, RNA polymerase when gets the termination signal, so the termination signal when it is reached, then the RNA polymerase will separate from the DNA molecule. If RNA polymerase is separated from the DNA molecule, so newly synthesized RNA transcript will also be separated. So RHO independent termination. So in some cases where the RHO is not functional, second GC rich region, so there are some regions known as GC rich regions where GC nucleotides are abundant in the quantity. So where they are in huge number, so this is making the loop like structure. So when the RNA polymerase is reaching at this loop like structure, so this loop is pulling mRNA molecule from the template DNA. In this way, the mRNA molecule is separated from the DNA molecule. Here we see that this is the DNA molecule the parental DNA molecule. So this was this is the promoter region. So means from this site the transcription process should start. So initially here the DNA molecule was melted, so the two sequences were separated, and on this one polynucleotide chain of DNA molecule, the process of transcription will take place. So it started from this region. So after when the process was over here, RNA molecule up to this point was separated from the DNA molecule, and then DNA molecule was rewinded, means reclosed, and here this chain is growing continuously, and here we say that this is the RNA polymerase molecule. This is growing and reading the nucleotides of the DNA molecule, and here it continuously binding the free nucleotides. So this is the direction of transcription from here in this direction, and here we see that the enzymes, these enzymes are melting the DNA molecule and separating the two strands. So we can see here that there are different sites on a single DNA molecule where the process of transcription can start in different directions. For suppose here direction is like this, at this site direction is like this, and at this side direction is like this. So here is the other strand, this one is other strand. So we can say the process of transcription can take place at very specific site of DNA molecule, single chain of nucleotides. Even it can take place on both chain of nucleotides, or it can take place at various sites, like here there are 3 sites where the process of transcription is started, while in case of replication, so replication is not like this, that it is not taking place at various sites. So it not is taking place at only small segment, but that one is a continuous process, and if transcription is if replication is started, so this will continuously go throughout the whole genome. So this is also the difference between the replication and transcription. So the transcription can take place at small segment of the DNA molecule, or at same time can take place at various sites of the same DNA molecule. Then we are learning about when the transcription is over, and after transcription, post-transcriptional modifications. There are some changes that are happening in the newly synthesized RNA molecule. They are called as post-transcriptional modifications. Endonuclease cleavage means there are some other enzymes. They are actually taking on the newly synthesized RNA molecule. For suppose poly A tail means at the end of transcription, the RNA molecule, on the RNA molecule, poly A tail will be added. So this tail can be from 20 to 250 base pairs. So this is depending in different species. The poly A tail size can be different. Even in prokaryotes and eukaryotes, the size of the number of poly A nucleotides can be variable, can be different. And the second thing which is happening, it is the addition of capping region. So in the beginning of the RNA molecule, a special type of nucleotide is added, which is 7-methyl guanosine triphosphate. So this region is added at the 5 end of the RNA molecule, and this process is known as capping. So capping and stalling both are very important for the newly synthesized RNA molecule. Then methylation of N6 of adenine residue and number 2 hydroxyl group of ribose done in cytoplasm. So this methylation is taking place in the cytoplasm, while this poly A tail adding and capping was done in the nucleus of the cell. Then what is happening? The intron regions of the DNA molecule, of the RNA molecule are removed, will be removed, and the exons will be added together. We will see this in this figure. This figure shows that this is the molecule that contains exons, introns both, and this is the DNA template strand. So when this DNA template is transcribed, means from this DNA molecule RNA molecule is synthesized. So this is the RNA molecule. So this RNA molecule contains exons, introns, similar like the DNA molecule. So after synthesis of RNA molecule, this molecule goes to the process of modification. What is happening in the modification? As we already learned that poly A tail is added in the end of the, or at the 3 end of the molecule, and at the 5 end of RNA molecule the cap region is added, 7-methyl guanosine triphosphate, the cap region, the cap nucleotide, this will be added at the 5 end of the RNA molecule. Then this is added. So this is known as modification. After modification there is another process known as splicing. So what is happening in splicing? Here at this stage the RNA molecule is non-functional, means that cannot be translated. So after splicing, this RNA molecule will be mature and will be ready to be exported out of the nucleus into the cytoplasm for translation. Means after that translation can take place on this mRNA molecule. So what is happening here in splicing? Actually introns, intron 1, intron 2, these both introns are removed, and exon A, B, C, they are added together. So these exons are actually the coding regions, they will be coded, while introns were non-coding regions, that's why they were removed from the RNA molecule. Now RNA molecule is mature. This is all about today's video. So hopefully see you soon. Allah Hafiz.